This presentation covers some of the most common solutions and equipment used in the culturing of mammalian cells, sometimes also referred to as tissue culture. Trypsin is an enzyme that cleaves proteins, and thus it detaches cells from their substrate by breaking the protein bridges cells use to adhere to the culture vessels. Fibronectin is the specific protein target cleaved by trypsin, and it breaks apart the protein at the amino acids lysine or arginine. EDTA is the abbreviation for the chemical that binds up calcium ions. These ions interfere with the activity of trypsin, so including it ensures that trypsin can do its job. Trypsin is not specific to fibronectin and eventually will attack other trypsin molecules and digest it in solution. Do not leave thaw trypsin for too long, or it will not be effective as it will have been digested up in your solutions. Similarly, leaving culture cells for too long in a trypsin-containing solution will reduce viability as the trypsin will attack other important cell proteins on the surface of the cells. Phosphate buffered saline, or PBS, is another important solution. This is an isotonic salt solution that is often used to wash cells to remove media and its components before a procedure involving the cells. Because calcium and magnesium interfere with some of the enzymes such as trypsin, which is often used in cell culture, it is important that the solution be free of these ions. As with any solution used on living cells, the solutions must be warmed so as to avoid shocking or damaging the cells. Trypan blue is useful when assessing the health of your cells or when calculating the number of living cells to passage to new flasks to perform an experiment. The diluted dye solution is mixed with a cell suspension for a short time before examining a small volume, such as 10 microliters, under a microscope. The total cell count and the percent of dead cells can be calculated because dead cells will be blue, but living cells remain colorless. Usually a 1-to-1 dilution of trypan blue solution is added to a dilution of cell suspension to do the observations. Propydium iodide is similar in function to trypan blue. However, this dye fluoresces red and binds DNA. Thus, when assessing living versus dead cells with the fluorescent microscope or with flow cytometry, propydium iodine is used in place of trypan blue. Common household bleach is the disinfectant of choice in cell culture. It's cheap and effective. With typical scenarios such as used in our laboratory, no special disposal procedure is needed. Simply add enough bleach to decolorize the medium. This will kill all the cells. After brief incubation of about 5 minutes, the waste can be rinsed down the sink. The flask can be disposed of in the trash after a bleach disinfection procedure. There are some basic equipment that every cell culture facility will have. This part of the presentation will go through them. One of the most important pieces of equipment for mammalian cell culture is the CO2 incubator. While insect cell lines do not need carbon dioxide in the medium, mammalian cell lines do require stringent conditions to grow, including 5 to 10% dissolved carbon dioxide in the medium. This environment mimics that of the body. CO2 incubators must be kept meticulously clean so as to avoid contaminating the cell lines growing within them. Should a culture become contaminated, a decontamination protocol should be employed before continued use growing more cells. A water bath or a dry bead bath will be used to warm media to the proper temperature, usually 37 degrees, before it's added to the cells. Again, this prevents cells from being shocked and damaged by extreme temperatures during laboratory manipulations. Oftentimes, dry baths composed of small beads are used in place of water baths. They take longer to heat the needed solutions, but they cut down the possibility of introducing contamination. Water baths are known to harbor fungi and bacteria and must be disinfected frequently, as well as have antimicrobials added to the water. The dry baths do not support microbial life as well, and thus reduce contamination. The media used in cell culture contains supplements such as fetal bovine serum and other growth factors. These contain heat-sensitive molecules that would be destroyed during processes that involve the autoclave. Thus, media is often sterilized using a filtration apparatus as shown here. A filter with a small pore size, such as 0.22 micrometers, is used. Liquid molecules can flow through the pores, but the filter traps bacteria and fungi on the surface, and media is collected in a sterile vessel below. Heat is not needed. Panel A shows this filtration setup, and B shows a colorized electron micrograph of bacteria trapped on the surface of a membrane with very small pores. The vacuum pump is another useful piece of equipment. Vacuums are needed to pull the fluid through the filtration apparatus shown previously. In addition, vacuums attached to sterile pipettes without cotton plugs are used to draw spent media into collection containers to be disinfected and then disposed. Of course, the used pipettes must be disposed of appropriately, typically into sharps containers or contaminated glass containers, so they can be disinfected without danger to the waste handler. The autoclave is an essential piece of equipment for any laboratory, including a cell culture lab. This instrument is used to sterilize instruments and glassware, media or buffers without heat-sensitive components, and biohazardous waste. The autoclave is basically a large pressure cooker. The chamber must reach 121 degrees Celsius. It uses steam under 15 pounds per square inch (psi) of pressure, and this condition is held for a minimum of 15 minutes to ensure sterility. The image at the bottom shows a common autoclave function verification system. A vial containing medium is placed within a larger, flexible tube containing heat-resistant bacterial endospores. This is sent through the autoclave. After autoclaving, the internal vial is crushed so that the medium contacts the endospores. Then it's placed in the incubator. An untreated control vial is also treated to release the medium, and both the test and the control are incubated. The unautoclaved tube should experience a color shift due to microbial growth acidifying the medium, but the autoclaved tube should remain unchanged because no microbial growth should occur. The most simple tissue culture hoods are laminar flow in design. These push filtered air out towards the experimenter, away from the work area. This design maximizes the protection of the cells in the work area. You can see this diagram that unfiltered air is drawn in at the top and then passes through a series of filters before being discharged down into the hood space and pushes out towards the experimenter. The laminar flow is different from a typical biological safety hood. Biosafety hoods protect the experiment and the experimenter. This requires a more complex design to control the flow of air. Air is drawn in from the edges of the hood, including one near the experimenter. This is sent through a high-efficiency particulate air, HEPA, filter before being directed down into the hood space. Air is vented out of the hood as well after it goes through another HEPA filter. Study these diagrams in order to understand this setup. Dead airspace enclosures are the most simple hoods. They may include a UV light to sterilize the interior before use. UV light also damages DNA, so this type of space is often used for polymerase chain reaction setups. The idea is that the hood minimizes air currents and dust from entering the culture. We made several do-it-yourself fashion for the initial micropropagation work in the classroom. There's a separate presentation on microscopes in the tissue culture lab. Please review it. Microscopes are, of course, extremely important when it comes to working with cells, which are microscopic and can't be assessed with the naked eye. The inverted phase contrast and the fluorescent microscopes used in cell culture may be new, but the typical upright Brightfield microscope is probably somewhat familiar, as it's the type of microscope used almost universally in many types of laboratories. Long-term storage of cell lines is accomplished using liquid nitrogen tanks. These tanks hold varying amounts of liquid nitrogen depending on size. Liquid nitrogen has a temperature of negative 196 Celsius, or negative 320 degrees Fahrenheit. A special freeze-down protocol must be followed to store viable cells. This type of storage is the only way to guarantee that cells will recover and grow once you thaw and replate them. This type of storage is used for stem cells, human sperm and eggs, and even human embryos at the earliest of stages when infertility treatments are being undertaken. This is the end of the introduction to the major cell culture equipment, although many labs employ specialty items and many smaller, more routine equipment items are used as well. I hope you're starting to become acquainted with this specialized laboratory working environment.